Hello and welcome dear students, professionals and learners to the video series on stability testing. So we have seen the basics of stability testing in the last video. Now in this video we will see the stability testing for new drug substances, and the guideline referred here is ICH Q1A, that is stability testing of new drug substances and new drug products. So in this video only new drug substances is covered, and in another video we will cover the new drug products. So let's start with the presentation. See, as we know that stability is required to provide the evidence on how the quality of the drug substance or drug product varies with the time under the influence of variety of environmental factors like temperature, humidity and light. So these three factors are considered while doing the stability study. These are temperature, humidity and light. To establish a retest period or shelf life and recommended storage conditions, this stability testing is required. Then stability data package for new drug substances or product that is sufficient for registration application within the three regions of EC, Japan and the United States. So ICH covers these three regions mainly and gives the guidelines for stability testing. The choice of test conditions defined in this guideline is based on the analysis of the effect of climatic conditions in the three regions of EC, Japan and United States. The mean kinetic temperature in any part of the world can be derived from the climatic data, and the world can be divided into 4 climatic zones, that is zone 1 to 4. This guideline addresses the climatic zone 1 and 2. So by this guideline we will understand the stability testing requirement for zone 1 and 2. Now you will have a question about mean kinetic temperature. So there is a standard criteria for this mean kinetic temperature. This mean kinetic temperature is calculated from the Arrhenius equation, and always remember that this mean kinetic temperature is not the average temperature. This mean kinetic temperature is not the average temperature, and as per my understanding from the literature, the mean kinetic temperature is slightly above that the average temperature. So based on this, the world is divided into 4 climatic zones. So ICH zone you can see in the blue region here. These are the ICH zone 1 countries. Then zone 2 is in the green color. This is green color. Then zone 3 is in the orange color, and very small dots are here. So zone 3 countries are very less from this picture, and zone 4 countries are red in color. Our India comes into this zone 4, and some of the Asian countries are also there in the zone 4. Then zone 1 is the temperate zone, zone 2 is subtropical zone or Mediterranean zone, zone 3 is hot and dry zone, zone 4A is again zone 4 is divided into zone A and zone B. So zone A is hot and humid, that is tropical, and B, that is hot and higher humidity. So let's see the countries. Zone 1 is Germany, European nations and Russia. Long term condition is 21 degrees Celsius and 45% RH. Zone 2. In zone 2, USA is there, Japan is there. So this, as per this guideline, USA, Japan and European countries are there. Australia is also in zone 2. Egypt, France, Greece, Japan. All these are having the long term condition of 25 degrees Celsius and 60% RH. Then coming to zone 3, very limited number of countries are listed in zone 3, which is hot and dry zone, and the countries are Iraq, Jordan, Botswana. Long term conditions are 30-35. So here temperature is more and humidity is less because this is hot and dry zone. Please consider these terminologies and remember these terminologies. Hot and dry zone, here temperature is more and RH is less. Then zone 4A, that is hot, humid, that is tropical. So Jamaica, Kuwait, Oman, Senegal, These are some of the countries. Senegal is an African country, just I have told to remember. So long term condition is 30 degree and 65% RH. Here temperature is increased with the increase in the humidity, but here in zone 3 temperature is 30, humidity is less. Zone 4B. So our country India is coming into this zone 4B. This is hot and higher humid. Long term condition will be 30-75. Then Brazil is there in zone 4B. Then India, Malaysia, Philippines. So these are the countries which are in zone 4B. Then what are the requirements of the stability study for new drug substances? So one by one we will see. First is the stress testing. This is also known as force degradation, where the drug is exposed to the harsh conditions that are above the normal stability conditions or above the formal stability requirements. The effect of temperature, humidity, hydrolysis, oxidation and photolysis or effect of light is considered in this stress testing or force degradation studies. So the drug substance or API is exposed to the harsh conditions to identify the degradation products and the pathways. Then to understand or to study the intrinsic stability of the drug. Intrinsic means what is the nature of the drug, at which type of force degradation is happening, under which condition it is happening, whether it is due to temperature, whether it is due to humidity, hydrolysis, light, acid base hydrolysis, by which of the factor the API is getting degraded, and based on that the intrinsic stability is understood. Then this stress testing is required to validate the stability indicating power of the analytical methods. Also, stress testing is done on the single batch of the drug substance. Once the drug substance is developed and now the applicant want to go for the exhibit batches of the drug substance or API, that time on one of the batch this force degradation study is to be done. This involves the study of effect of temperatures in 10-degree increments. If you know that at accelerated temperature 40 degrees is accepted by the regulatory authorities, but here in the force degradation study or stress testing it is done at 50 degree, 60 degree and sometimes above that. Then humidity is 75% or greater. Then hydrolysis across a wide pH range values which involve the use of acid and bases. Then oxidation, generally peroxide is used for oxidation purpose and light is used for photolysis studies. Then coming to the formal stability testing, like the drug product stability testing, API also has to show the formal stability testing and the data for approval. That may be DMF, ASMF or other type of drug master file application for the approval of the API. So number of batches are at least 3 primary batches. So what is the meaning of primary batches? These are the batches which are representative of the commercial scale. The batch size may be less, but the process and equipments are for the commercial scale. So scale is at least pilot scale. Then manufactured by final method or final proposed method, and the method of manufacturing is representative of the commercial manufacturing. Once you apply with these batches and scales and methods, then it will become fixed for the approval reasons. Then container closure. The container closure should be same as or it should simulate the packaging proposed for storage and distribution. So all these are the things like the manufacturing method, container closure and the specification. These are finalized by the R&D department during their research, and then with these methods and specifications and container closures and everything, this goes to the plant whether the manufacturing is to be done, and there the 3 pilot scale batches are taken for regulatory approval purpose. So we have seen number of batches, scale, manufacturing process, container closure requirements. Now we will see the specifications. So as like for the drug product, safety efficacy is much important. So the same is applied here. And that's why the specification includes physical, chemical, biological and microbiological attributes to be studied, as these affects the quality, safety and efficacy. Then the analytical methods whichever are applied to the final batches that are required to be validated, and this should be stability indicating. Stability indicating means these methods, like if you consider the assay, that assay method should disregard the impurities and only should consider the API, so that you will get the actual potency values of the API. Then coming to the testing frequency. So all of us know that three conditions are there for testing: long term, accelerated and intermediate. So for long term stability, this is to establish shelf life. At least 12-month data is required for long term stability condition. Frequency is every 3 months over a first period. First period, that is first year. So every 3 months it will become 3, 6, 9, 12 months. Then every 6 months after the second year it will become 18 months and 24 months, like that. Then it will become annually from the third year, or every 6 months over the second year, then actually thereafter through annually through the proposed retest period. So whatever the retest period is there, whether it is 4 year or 5 year, you should check the API annually. Then accelerated storage condition. This is required to extrapolate shelf life, and the total time period is 6 months. So the frequency is 3 months and 6 months. If the API is prone to the degradation at this stage, then additional time points can be included, and you can have the frequency like 0, 0, 1, 2, 3 and 6 months, or you can have one time point between the 3 and 6 months also. That is also okay. If the significant change is expected during the accelerated stability study, that means you have to include fourth time point. Then coming to the intermediate storage condition, this is the storage condition kept if there is a significant change at the accelerated storage condition because of this higher temperature like 4075, then we have to consider this intermediate storage condition. That is, minimum of 4 time points should be there, including initial and final time points. Example is 0, 6, 9 and 12 months. At the submission time, 6-month data is required from the 12-month period. So for long term condition, it is till the retest period or shelf life period. Intermediate will be 12 months and accelerated will be 6 months period. Then storage conditions. See, we have to check the thermal stability and sensitivity to moisture for the API. That's why this storage condition and duration should be sufficient, so that the storage, shipment and subsequent use period can be covered. For this, long term is 12 months and continued for a period of time sufficient to cover the proposed retest period. Data from the accelerated storage condition, if appropriate from the intermediate storage condition, can be used to evaluate the effect of short-term excursions outside the label storage conditions, such as might occur during the shipping. So the main is long-term condition, and if the product is studied at long term, that should cover the retest period. Then storage conditions. The long term, intermediate and accelerated are given. So long term is 25 degrees Celsius temperature and 60% RH. It is known as 2560 or 3065 for 12 months. Then intermediate, this is 3065. The time is 6 months. Then accelerated, This is 4075 and it is 6 months. Generally, by the applicant, this long-term and intermediate study is done for 12 months to have better understanding of the product. And long-term is also done at that period of time which is to be given as a retest period. So up to 5 years, this long-term condition is studied by some of the applicants. So long-term condition, it is up to the applicant to decide whether the long-term stability condition is performed at 2560 or 3065. And if you perform long-term stability study at 60 at 3065, then intermediate condition will not be there. Then coming to the drug substances intended for storage in refrigerator, this condition is referred as 2 to 8 temperature. So long term will be 2 to 8, that is 5 degree plus minus 3 degree Celsius, and time is 12 months. And for these type of drug substances, the long term condition is the accelerated condition. If you consider here, that is 25 degree and 60% RH. This is the long term condition for stable products or stable APIs. But if the drug substance is prone to degradation due to temperature, the long term condition becomes 5 degree and accelerated become 25. So your room temperature will become the accelerated condition. If significant change occurs between 3 and 6 months, testing at accelerated storage condition, the proposed retest period should be based on the real-time data available at the long-term storage condition. Then, if significant change occurs within the first 3 months, testing at accelerated storage condition, a discussion should be provided to address the effect of short-term excursion outside the label storage condition, that is during shipping and handling. So this is nothing but the effect of temperature. What is the sensitivity of the API to the temperature? That is required to be studied and justified. Then coming to the drug substances intended for storage in freezer. So long-term condition is minus 20 degrees Celsius and there is no accelerated condition. For drug substances intended for storage in freezer, the retest period should be based on the real-time data obtained at the long-term storage condition, that is minus 20 degree. In the absence of accelerated storage condition for drug substance intended to be stored in freezer, testing on a single batch at elevated temperature, that is 5 degree, that is 2 to 8. So 2 to 8 will become the elevated temperature, or 25 degrees Celsius will become the accelerated or elevated temperature for an appropriate time period should be conducted to address the effect of short-term excursions outside the proposed level condition, that is during shipping and handling. Now see, if the drug substance is stored at minus 20 degree and there is a slight increase in the temperature, so we have to study the impact of elevated temperature for the drug substances intended for storage in the freezer. Drug substances intended for storage below minus 20. So some of the APIs may be there which require deep freezer for storage and their temperature is minus 20 below. So drug substances intended for storage below minus 20 degree should be treated on case by case basis. So this will depend on if the applicant have all the data for the 3 batches, then no commitment is required. If long-term stability data on primary batches is not covering the proposed retest period granted at the time of approval, then commitment by the applicant to continue the stability studies post approval to establish the retest period. The simple meaning of this is that if the applicant is not having the complete data for stability, then applicant will give the commitment that they will continue the stability study till the retest period is completed, so that the retest period is firmly established. So 3 points are there. If submission include data from stability studied on at least 3 production batches, a commitment should be made to continue these studies through the proposed retest period. Suppose you are giving a proposed period of 5 years, so continue the study till 5 years. If the submission includes data from stability studied on fewer than 3 production batches, that means 2 batches are taken on pilot scale or 1 small scale batch, or so 3 of the pilot scale batches are there, but these are not the production scale batches. Then a commitment should be made to continue the studies through the proposed retest period and to place additional production batches to the total of at least 3 on long-term stability studies through the proposed retest period. So this is the commitment requirement from the applicant. Then criteria 3. If submission does not include stability data on production batches, a commitment should be made to place the 3 production batches on long-term stability studies through the proposed retest period. So whenever there will be batch manufacturing for production scale, so for 3 batches, the applicant will study the long-term stability study throughout the proposed retest period. Suppose the retest period given by the applicant is 3 years. So 3 batches on production scale should be loaded on stability for 3 years. Then stability evaluation. The stability data of 3 batches to be studied. Whatever the data is there for the 3 batches, that is required to be studied in detail, and the degree of variability of the individual batches affects the confidence that future production batch will remain within the specification throughout the assigned retest period. The simple meaning of this is that whatever the data is available for the 3 primary batches or stability batches, if that data shows the within batch variability, then there will be a less confidence for the future production batches. And if that data is very good, that means meeting to the specifications without any variability within the batches or within the time points, then it will give the confidence to the applicant as well as the regulatory authority that the future production batches will remain within the specification throughout the assigned retest period. If there is a degradation at some of the batches, at some of the conditions, if variability is there in the data, for example, say assay water content or related substances or impurity, so this variability will decrease the confidence. Now the data may show a little degradation and so little variability, that is apparent from the looking that the data at requested retest period will be granted. Simple meaning of this is that if the data is well within the specifications limit and there is a little degradation or little variability, so it will be suggested that the requested retest period can be granted to the applicant. If there is a variation in data among the batches and if degradation is observed, then principles of extrapolation and statistical analysis are required, and these are employed to apply for the extrapolated shelf life. Sometimes based on this extrapolation, the shelf life can be granted, and sometimes the applicant is said by the regulatory authority that you first submit the long-term data and based on that your drug substance will be approved with the final shelf life, and based on this retest period can be given and justified. So after getting the total data in our hand, a storage statement and labeling is done based on the stability data. So a storage statement should be established for the labeling in accordance with the relevant national or regional requirements. The statement should be based on the stability evaluation, that is based on the stability data of the drug substance, and where applicable, Specific instructions should be provided, particularly for drug substances that cannot tolerate freezing. Some drug substances cannot tolerate freezing, and these type of statements and instructions should be clearly provided by the applicant. Terms such as ambient condition or room temperature should be avoided, because these statement doesn't give the exact idea of temperature. So temperature should be included in the storage statements. A retest period should be derived from the stability information, and retest date should be displayed on the container label if appropriate. So if you don't have expiry period from the stability data and you are giving a retest date based on the available stability data, then that retest date is required to be displayed. So I hope you might have got some understanding out of this video. This is for new drug substances, and another video is there for new drug products, that means formulations. For this I have referred mainly the ICH guidelines and other guidelines related to the stability studies. Thanks for watching this video, and keep watching the video, as many of the people don't understand the terminologies of these guidelines, and through these videos you can get more clarity for the topics. Please do like, share and subscribe to the Pharma Learning in Depth. Thanks for watching.